Hello everyone and welcome to our third mini lecture from the alcohol unit. Today we're going to be talking about alcohol's effects on glutamate system. So for those of you who don't recall, glutamate is the principal excitatory neurotransmitter of our nervous system. It's an amino acid, it's very ubiquitous, and it does all kinds of excitatory signaling. The greatest effects of alcohol on the glutamate system take place at NMDA receptors. So as you may recall from our extensive discussion of long-term potentiation, that NMDA receptors govern an ion channel that is permeable to calcium and sodium, making it very, very important for things like learning and memory and synaptic plasticity. Alcohol serves as an antagonist at this receptor, so it decreases the effectiveness of NMDA receptors. In addition to its effect on NMDA receptors, alcohol also reduces the release of glutamate in many areas. In particular, we know that this reduces glutamate release in the hippocampus. Hippocampus being a brain region that's very, very important for learning and memory. So temporary inhibition of NMDA receptors all over and reduced glutamate transmission, so reduced glutamate release, can sort of work together and may contribute to amnesia. So the experience of having had too much to drink and forgetting events or entirely blacking out is most likely attributable to the effects at the glutamate receptor, at the NMDA receptor and reduction in glutamate release. So in addition to those effects that take place during the actual sort of binge intoxication state, there are pharmacodynamic changes or forms of tolerance that take place with repeated exposure. So, kind of unsurprisingly, NMDA receptor upregulation takes place, right? As a compensatory sort of mechanism, because NMDA receptors are being antagonized, we see an increase in the prevalence of NMDA receptors with chronic use. Additionally, during withdrawal, we see a rebound effect in terms of glutamate release. So because glutamate release is reduced during chronic use, the rebound state is an increased release of glutamate. So during withdrawal, you see this rebound where the amount of glutamate release just sort of bounces back above baseline. This results in a hyperexcitable state that can cause seizure activity in alcohol abstinence syndrome or in a withdrawal state. This actually works in combination with some changes that take place in the GABAergic system, which we're going to talk about next time. So a combination of these can cause excessive calcium influx, which can contribute to cell death. If you remember in our glutamate and GABA unit, we talked about how excessive stimulation of NMDA receptors can cause excitotoxic cell death. Again, because of the combination of these two things, increased release of glutamate in a withdrawal state and an increased amount of NMDA receptors is sort of a perfect storm for one, hyperexcitability, and two, excitotoxicity from overstimulation of NMDA receptors. So you can see over here on these graphs on the left, these are rats that were made dependent on alcohol and then were put into a withdrawal state. So here you see glutamate output, and this is the number of hours since they were taken off of alcohol. You can see here in red the alcohol group. During alcohol withdrawal, you see this spike in glutamate output that sort of tapers off over time. In blue, we have a sucrose control condition where they were administered sucrose instead of alcohol. You can see that withdrawal from sucrose produces no such change. Down here is a subjective behavioral withdrawal score, and you can see that the withdrawal score tracks really nicely with this glutamate output change, suggesting that they are related. Okay, that's it for the glutamate system in alcohol. We'll see you next time.